So radiolarians live in marine environments. They're heterotrophic. They eat algae and bacteria. They have pseudopodia. Sometimes they have an algal symbiont, so they have an organism living within them that helps produce energy from sunlight. Radiolarians produce a shell made out of silica. That's important because there are other protists that make shells as well, but radiolarians make their shell out of silica. Historically, seabeds that are covered in radiolarians get compressed and result in the formation of a mineral type called radiolarian chert. You can see here, this is a hill outside of San Francisco, these layers here, where at one time millions and millions and millions of radiolarians piled on top of each other, compressed, and now they form a mineral type we call radiolarian chert. That radiolarian chert is actually made up of those shells. And these shells are really fantastic. They have a bunch of different morphologies. Some of them look like fidget spinners or ninja stars or bicycle gears or weird helmets. And they really are cool. One thing to note is that radiolarian shells oftentimes have spikes or points, and they look kind of sharp and like they might not be too fun to touch. So if you see an example of a shelled organism, a shelled protist, and it's spiky like that, it's probably a radiolarian. Another possibility, if you find a protist that has a shell, is that you're looking at a foram. Foramiferans also form a shell, except they form their shell out of calcium carbonate. You can see that these shells tend not to be spiky. They tend to be pretty round and like something that you could hold in your hand and it wouldn't hurt you necessarily. So if you see a tiny microscopic protist, maybe under a microscope, and you see a shell and it's not spiky, it's pretty round, there's a good chance you're looking at a foram. Forams are heterotrophic. They also feed on algae and bacteria, and they move by a pseudopod. So they have a foot, kind of like an amoeba, can stretch and move in and out of that shell. Foramiferans form a foundation for marine ecosystems, and without them, those ecosystems would likely collapse. In order for forams to survive, they have to form these calcium carbonate shells. Well, the formation of those shells is highly dependent on a balance of pH in the water where they're found. As CO2 levels rise with human impact, there's more CO2 in the air interacting with the surface of the ocean. And as that interaction happens, that CO2, of course, is absorbed into the ocean, and that changes the pH. It lowers the pH, and it creates a more acidic ocean. You may have heard about ocean acidification and the implications of that, but one of the implications of that is that forams, which is foundational in those ecosystems, would have trouble creating those shells. Corals also form calcium structures, and to do that, they also need this delicate pH balance. In this particular experiment, they introduced CO2 into the aquatic environment, which lowered the pH. As that happened, there was lower fertilization rates, and there was also a reduction in recruitment and settlement of those corals. So with corals, we can already see that there's an impact from CO2. Corals are having a really hard time all over the world. We're experiencing bleaching events, which used to be fairly uncommon, but now we're seeing bleaching events happen in rapid succession year after year on larger and larger scales. And the issue with bleaching events is that whenever they happen, it takes a really long time for those corals to recover. If they continue to happen in rapid succession, which is likely to happen as ocean temperatures rise due to global warming, we're likely going to lose a lot of coral systems. And that's just by the coral organisms leaving the coral. There's also the issue with them not being able to form the corals themselves as that acidity drops. Here's an example of elkhorn coral completely bleached. And those microorganisms that live in the coral have left. And so what's left behind is just this calcium carbonate shell, this calcium skeleton. So what does it have to do with forams? Well, maybe that's kind of a glimpse into the future of forams. if CO2 continues to drop the pH of our oceans, we might experience a collapse of marine ecosystems because forams will be affected. Has this happened before? Recent research has suggested that one of the major contributors to extinctions in the oceans after the impact at the Chicxulub crater, the one that killed off the dinosaurs, one of the major contributors to extinction may have been the collapse of the marine ecosystem because of the collapse of the foram community. In this graph, you can see that the diversity of forams in the fossil record, it stops abruptly right at the time when that meteor impacted. You can also see evidence of a drop in pH. And finally, you can see evidence of a rise of CO2. So in effect, you have a very similar process here where CO2 went up, pH went down, and foram diversity collapsed. Now this happened near the surface of the ocean, and that's where you saw these marine extinctions. Down in benthic systems, deep underwater, there wasn't a big change in pH, and you also didn't see as many species go extinct. That's why a lot of these living fossils, these species that were around during the dinosaurs, continue to exist at those very, very deep layers of the ocean.